Creating words. Much of what you're learning this semester is essentially a new language of microbiology. We are creating words using pieces and parts, often derived from Latin or Greek terms. Common suffixes that we can use: phile, meaning loving; troph, referring to a nutrient requirement; tolerant, just as you would imagine, can grow in the presence of or can tolerate. And an example of creating words by putting parts together is the word extremophile. Today, almost everyone knows what the word extreme means. It means it's sort of above and beyond the average or the typical. An extremophile is, of course, something that loves those extreme or above average conditions. Terms that we use to describe temperature requirements, we'll use that suffix phile and add a prefix or root word that refers to temperature. A psychrophile is a cold-loving type of microorganism. Now, the illustration of the graphic representation of temperature requirements for the different categories will change a little bit from one resource to another, but roughly -4 degrees to about 30 degrees Celsius would be a typical growth range or temperature range for many psychrophiles. In other words, they grow better in the refrigerator than they might, let's say, perhaps at room temperature. This is why your food spoils even in the refrigerator. There are psychrophilic bacteria and microorganisms that can be in many foods or on many foods, and they're continuing to grow and grow even more rapidly in those colder temperatures than at warmer temperatures. Mesophiles are microorganisms that grow at the more likely body temperatures or room temperatures that humans prefer, anywhere from 15 to 45 degrees Celsius. Notice that for each of these graphic representations, psychrophiles, mesophiles, and the other categories like thermophiles, we have a minimum lower temperature, which for thermophiles, for example, is 40 degrees Celsius. We have an optimum, which for thermophiles would be roughly 65 degrees Celsius. And then we have a maximum, which again for the thermophiles would be 80 degrees Celsius. If you compare the temperature range of a thermophile to a mesophile, you can see there's a tiny little bit of overlap between the temperature maximum of a mesophile and the temperature minimum of a thermophile. But notice that thermophilic bacteria are not going to grow at room temperature. Mesophilic bacteria are not going to grow at those hotter temperatures of the thermophile. And in fact, any of these categories, the organisms just won't grow at the optimum or the highest temperature, and they certainly won't grow at the average temperature or optimum where the graph peaks at the top. So all of these categories for temperature requirement can limit or can prevent the growth of other categories of organisms. This even includes the hyperthermophiles. The hyperthermophiles are different in terms of temperature tolerance than the thermophiles. The thermophiles live in really hot conditions, but the hyperthermophiles, they can grow in even temperatures that reach slightly above boiling water. So we have roughly 65 to 110 degrees Celsius temperatures for hyperthermophiles. Some examples of thermophilic organisms would include some archaea that were first isolated in the 1960s in smoldering piles of coal dredgings or coal remains from mining industries in Indiana in the United States. Now for archaea, they don't have peptidoglycan if they have a cell wall. If an archaea has a cell wall, it's going to have what we call pseudo-peptidoglycan. So archaea in general have pseudopeptidoglycan if there's a cell wall present. But for Thermoplasma acidophilum, which is our example thermophile, this archaea has no cell wall at all. It's simply a cell wall-less archaea. Now, it does prefer temperatures at 55 to 60 degrees Celsius. Compare that to the average body temperature of about 37 degrees Celsius. So that's pretty hot for us, but it's ideal for Thermoplasma. And notice that in the name of the genus of this organism, it hints to us, or suggests to us, that this is a heat-loving organism, Thermoplasma. Another key clue to the growth conditions required by this organism is the species name, Acidophilum. It really prefers, or as the suffix implies, phylum, it loves acid environments with a pH range for growth of 0.5, which is extremely acidic, to 4. So if you've ever heard of acid rain or acid drainage from coal mining, this is what we're talking about, the extreme acidic leaching materials from those coal piles and the heat that's generated when they sort of have this extra sort of smoldering ignition that can occur in this combustible coal is the ideal, really hot, acidic growth condition for Thermoplasma acidophilum. There's a photo in the upper right-hand corner of a close relative, Thermoplasma volcanium, and that particular species, volcanium, actually grows in and around volcanoes. It'll grow in those smoldering, acidic, extremely, for us, challenging environments, but Thermoplasma volcanium can grow quite nicely in and around those volcanoes. If we take a look at other places in the world, we might see the same types of Thermoplasma growing in those hot, acidic environments. And there's an image of Thermoplasma acidophilum in the lower right-hand corner. You can see it doesn't have that typical geometric shape that we describe for most prokaryotes. It's sort of an amorphic looking thing, or very amorphous and irregular in shape. And it does have one of the smallest genomes ever sequenced. So it's a tiny little organism, highly uniquely adapted for growth in those hot environments. We just mentioned pH, or acid environments for Thermoplasma. We have the temperature requirements, we have the pH requirements. An acidophile is an organism that is going to prefer or loves to grow in an acid environment with a pH below 7. Cephalobus is an archaea that can also be found growing in and around volcanic hot springs. It prefers a pH of roughly 2 to 3, and notice that for acidophiles, a pH range can be anywhere from 1 to roughly 5.5 or 6. Again, depending on the resource of the textbook, these ranges of sort of growth requirements can differ a little bit, but they're usually pretty close. We have a pH minimum, we have a pH maximum, and we also have an optimum pH for the acidophiles. Same thing's true for the next category, which would be the neutrophiles, and the neutrophiles prefer a neutral pH. The alkalophiles are the third category of pH requiring organisms, and those organisms prefer to grow at pH above 7. Our classic example of an alkalophile is Vibrio cholera. We've already mentioned Vibrio cholera when we described some general characteristics of prokaryotes in terms of shape. It's a comma-shaped cell, and flagella. It does have essentially a single flagellum. If we look in the soda lakes of Africa, we can also find another alkaline-loving or above pH preferring bacterium called Natronobacterium. In the soda lakes in Africa, which have a very alkaline pH, we can see from this aerial photograph the pink color of the lake. Most of the time when you think of a lake or some kind of body of water, you expect to see some kind of blue color. But notice for both this acidophile of Cephalobus and the alkalophile of Natronobacterium, we see different colors. These very extreme conditions and these types of organisms have adapted and produced various kinds of chemicals in their cells that take advantage of these unique growth conditions, and as such they have some unique colors that we see that tells us something's different about what's growing in this water. So when you see those colored types of materials in various water systems, the first thing you might want to think about is maybe that's the microorganism itself that's lending the color to these water systems, which is exactly what we're seeing in these photos. Salt, we use the prefix halo or natro, which we've just seen in the previous slide for the alkalophile, but now we're going to also include, what does that mean? It's referring to salt. So Natrobacterium not only likes the alkaline, but it also likes salt. Halotolerant organisms can grow in the presence of a high salt concentration. High salt concentration provides a high osmotic pressure on the outside of any cell that's growing in that very salty environment. A halophile actually requires a very high salt concentration for growth. For halotolerant examples, we have Staphylococcus aureus. Staphylococcus aureus is a very common organism to find growing on our skin. If it gets into the wrong place at the wrong time, though, Staphylococcus aureus is a frank pathogen that does produce chemicals that we call virulence factors, which we'll talk more about in another lecture. Those virulence factors can make us sick. Unfortunately, Staphylococcus aureus, when it does get into those wrong places at the wrong time, can grow in a wide variety of environments that are often found in and on the human body, including where we might have some high salt concentrations. In fact, that salt tolerance of Staphylococcus allows it to grow very nicely in, let's say, if it's getting into our food. Staph aureus is a common cause of food poisoning, and even if you have a lot of salt in that food product, Staph aureus can probably continue to grow because it can grow in the presence of up to 7.5% sodium chloride. In fact, one of the augers that we use in lab to isolate and identify if a Staphylococcus species is present is something called mannitol salt agar that has a 7.5% sodium chloride content. Staph aureus will easily grow and show up on MSA or mannitol salt agar. For halophiles, we're going to categorize those as either slight, moderate, or extreme, depending upon just how much salt